we are taking our last part of the course. Today we're starting with a biotransformation, and then I think next week we should have our last lecture for the course, so that you can have the rest of the time to prepare for your exams. Biotransformation for you, we'll take it as in terms of pharmacology, because you are doing biomedical sciences. Now, I say so because generally you have pharmacology divided into pharmacokinetics and pharmacodynamics. If you were maybe some environmental scientists or say toxicologists, similar things, we'll refer to them as we'll refer to it as a toxicokinetics and toxicodynamics. But in your case, we'll look at it as in the field of pharmacology. And so, as I was saying, that you have pharmacology divided into pharmacokinetics and pharmacodynamics. Pharmacokinetics simply refers to the activities of the body on any xenobiotic, any external substance that is taken into the body, any medicine, a drug that enters into the body. How does the body act on these drugs? And these processes are divided into 4 main stages, which are absorption, distribution, metabolism, and excretion. And so you find that pharmacokinetics is often referred to with the acronym ADME. That is the first letters of these stages: absorption, distribution, metabolism, and excretion. As a matter of fact, the name which is given to this particular part of your course as biotransformation actually refers to this third stage, which is metabolism. So metabolism of drugs is the same as biotransformation of the drug. Now, what it means is that you have a drug that gets into the body, and the absorption will be dealing with how the drug will get into the body, the route of entry. Distribution will be after the drug gets into the body, how it gets distributed to the various organs and tissues. And then the biotransformation itself, which occurs mainly in the liver. And in the end, the drug will get eliminated or excreted out of the body, either metabolized. So it means that there will be some metabolites of the drug, or the drug gets excreted unmetabolized. A lot of times you have both the unmetabolized drug and its metabolites getting excreted in different amounts. And you have the pharmacodynamics, referring to the activity of the drug on the body. So here you look at the mechanism of action of the drug. We did look at cell communication, and so here too you see the interaction of the drug with receptors on the cell surface, or even receptors that are inside the cell that the drug can pass through the cell membrane. These will be some lipophilic drugs that can actually pass through the cell membrane into the cell and go interacting with the receptors inside the cell. We also look at what we refer to as dose-response relationship. That is how the drug affects the body based on the amount of which is taken into the body. And then take a look at it's a drug, but it can still have some toxic effects depending on the concentration of it, the amount of it which is taken, and its concentration in blood. It can have some toxic effects. So take a look at therapeutic effects and then toxic effects, comparing these things. So generally, these are the things that we'll be looking at under this part of the course. Today, what we'll do is we'll take pharmacokinetics and go through these stages: absorption, distribution, metabolism, and excretion. We'll take, in order not to make it boring, we'll take some specific examples of some common drugs and look at their metabolism, because specifically this part of the course is on biotransformation. So we want to see how a medicine which is taken in can break down, going through various metabolic pathways. And then next week, when we come, then we take the pharmacodynamics to seal the course for the semester. So let's dive in. Now, as I was saying, if you are exposed to any drug substance, that drug substance gets into the body. So if you take the rectangle as inside of the organism, and the outside of it, outside of the rectangle, is its environment. But in this case, don't pay so much attention to its environment anyway, because it's not like you are being just exposed and the thing is acting on you. That is, if it was that you are looking at it in terms of toxicology, you can look at it that way. But this one is medication, so you are actually being administered the medication. And so absorption refers to the process by which the drug gets into the body. And then the process of distribution. After the blood gets into the body, it gets into the bloodstream and it starts to move to various organs and tissues. That's distribution. Now, when it gets into the body and starts to get distributed in the bloodstream, there can be some of these drugs that bind to even the blood proteins, like albumin. You'll find some drugs that can bind to blood proteins. And you also find some of the drugs that when they get to the target tissues, they will first get bound onto the cells, and then they have their effect on that cell. Or from distribution, you can have biotransformation of the drug, which is what we are referring to as metabolism of the drug. So it means that it is the metabolite of the drug after biotransformation, which will then have effect before there is excretion of the drug. Or even after biotransformation, you can have the metabolites also binding onto some specific cells or some proteins, and then you can have its therapeutic effect. In some cases, you can have from distribution directly without any biotransformation, the drug itself directly is having an effect before it gets excreted out of the body. So you can have the drug, going through these various ways and having its therapeutic effect. Now we take the process of absorption. So this is what we are doing. I said we are taking mainly pharmacokinetics today. So what we'll do is we take absorption, and then when we finish, we'll take distribution. Then we move on to the metabolism, which is the biotransformation of the drug. And then after that, we move to the last stage, which is excretion. And then then take a look at some other aspects that we can consider in terms of drug metabolism, even pertaining to metabolism in the environment, we'll take a look at some of those things in addition. Now we see there are different routes of entry that are depicted on the diagram you're seeing here. The first is oral, which is the most common form of drug administration through the mouth, and that goes into the gastrointestinal tract, the digestive system. And from the digestive system, you have the drug getting absorbed in the small intestines. The drug will get, just like the way when you take in food, the food gets digested, and when it gets to the small intestines, you have these nutrients being absorbed through the villi into the bloodstream. Same thing you are seeing here, that you have in the gastrointestinal tract. You have the drug which is taken in orally, getting absorbed in the small intestines into the bloodstream. Now in the bloodstream, it starts to flow to different organs. And as we just talked about, that digestive system where you take in food, the food goes from your mouth through the esophagus to the stomach, and then you describe all those processes, what happens in the stomach, the duodenum, small intestine, all the way large intestine, and all of that. Here too, when you take in any xenobiotic, any chemical substance, where biotransformation occurs is the liver. So as the drug moves with the bloodstream, it gets to the liver, that organ which is responsible for the metabolism of the drug. And as metabolism occurs there, you have the metabolites and the drug itself getting stored in the bile. What the liver does is we can refer to it as detoxification, even though it is not all the processes of metabolism of the drug which will result in detoxification, because in some cases you have the metabolism of the drug rather resulting into toxification of the drug. So that's the metabolites of the drug rather becomes even more toxic than the drug itself. But the main purpose for which, the main purpose intended for the liver acting on the drug is to get the drug out of the body, because it's a foreign substance. It's not a substance which is synthesized by the body. So the body, has to get rid of it. And the system by which it gets rid of it mostly is the liver. And what the liver will do is to transform the substance into some form that can be excreted out of the body. And one of the ways it does that is for it to now store it in the bile, in the gallbladder. We saw that structure looking at the gallbladder, which was embedded in the liver. And from the gallbladder, you have the bile, which is excreted into the duodenum, which means back into the gastrointestinal tract. And remember, bile is excreted into the duodenum to emulsify lipids, the fats and oils that are in foods. And once that happens, it means that excreted, portion of the drug, which is with bile, eventually gets excreted through stool. We also have some drugs which are taken in by inhalation, so through the nose. And so those drugs will then go into the lungs. And in the lungs, they get into the small pockets of the lungs, what we refer to as the alveoli, which are packed with blood vessels, or say capillaries. And then you have the the drug substance getting absorbed in the alveoli into the bloodstream. Eventually, the excretion of that drug is by exhaled air, the air that you breathe out. You have some drug, some substances getting out of the body by exhalation. You also have the intravenous route of administration of drugs. So these will go directly into the bloodstream, and from there it can go into the lymph, which has a role to play in the immune system. And this goes to the kidneys, where you have filtration, and formation of urine. Then urine is stored in the bladder, and so the drug substance is excreted out of the body through urine. From the blood lymph, you can also have some extracellular fluid, like plasma, flowing, which you have the drug flowing with it. And these can now get excreted by the exocrine glands, such as the sweat glands in the skin, which means that these drugs will get excreted through sweat. So you can have drugs getting excreted through sweat. You can also have drugs that, as extracellular fluid flows to different organs. These can go into, soft tissues like cartilages, and you have some of it getting into the bones. And this particular aspect is interesting in a way that there are some toxic substances that can get stored in the bones, and as if they are not there. Also the same thing, you can have some getting stored in the adipose tissue, which will mean that those substances are lipophilic, and so they get stored in those fat cells under the skin, and it will be as if they are not there. And it is only in old age or say in sickness, when such a person begins to lose weight, you have these substances that are getting absorbed back into the bloodstream from the adipose tissue and from the bones. You do have also some other route of administration of drugs, like the cutaneous, subcutaneous. So these ones will go into the skin or under the skin. You have the intramuscular. So this will be directly into the muscle. And you have intracavity. So these drugs, these ones are by some specific procedures that you have these drugs delivered directly into some cavities of the body. But eventually this will end up in the blood and also come through the various excretion pathways like urine, sweat, the soft tissues, the bones, and the adipose tissue. Now, when you take a look at that process of absorption and you represent it with a kinetic curve such that you have concentration on the horizontal axis over time on the horizontal axis. So you have concentration on the vertical axis, time on the horizontal axis. If a drug is taken in orally, with a process that we just described, that first the drug goes into the digestive system before it starts to get absorbed into the bloodstream. Now, what happens is, as you look at this, let's say if you take this first curve, you realize that you have the concentration of the drug rising steeply all the way. It gets to a certain maximum, and then the concentration begins to fall over time all the way and becomes consistent, and then later all the drug is then eliminated out of the body. Now, what does this tell us? When you take in a drug through oral administration and the drug begins to get absorbed into the bloodstream, the concentration will be increasing. And as the concentration increases, it gets to a point, the maximum. And at that point, you should also take into consideration that even as the drug is being absorbed at the same time, the drug is getting distributed to various organs, various tissues. And at the same time, when the drug begins to get to the liver, it will start undergoing metabolism. So the concentration of the drug will begin to reduce. So that's where you see there's a maximum, and then you see the drug is getting reduced all the way. And at a certain point, the drug now will start to get eliminated out of the body as a metabolite or unmetabolized drug. And so you see that gradual removal of the drug excretion from the body until you have zero concentration. But usually, drugs are administered with multiple dosages. So, apart from some which are taken, say maybe just a single dose, like some of these dewormers that they just give one, but many of these drugs that are prescribed, you will attest to the fact that they are of multiple doses. Like either you take one every 4 hours, or they tell you take one morning, take one afternoon, take one in the evening after meals, or one morning, one evening. So you have these multiple doses. Now, if it is so, then it means that this curve will look more like repeated over a certain period of time. So that if you are looking over a period of, say, 96 hours, what you see is that just as the drug reaches a certain point of elimination, another dose is taken, and so that it increases the concentration again, and the curve repeats. And the same thing happens third time, fourth time, which is to say that the concentration of the drug is being kept at a certain range. And the reason for that actually is for the drug to maintain its therapeutic effect. But what you see is that you have a low point, which is labeled C minimum, which is to say that is the minimum concentration that will have effects. So the minimum effective concentration. And you have an average, which is an average of that minimum and the maximum. That is the highest point, the highest concentration that the drug has reached. And so what we do refer to this, the distance between the minimum effective concentration and the maximum therapeutic concentration. That distance between them, we refer to it as a safety corridor. A safety corridor in the sense that if the concentration of that substance should go above that maximum concentration, then that substance begins to have a toxic effect. And so it is within the range of that interval between the minimum concentration and the maximum concentration that the drug will be having its therapeutic effect. If the concentration is below that minimum effective concentration, the drug will also not be having its intended therapeutic effect. So at least that minimum is required for the drug to have its therapeutic effect. Right. How about when you take in drugs through intravenous administration? Now, you see, when you have drugs introduced into the body through intravenous administration, what it means is that the substance goes directly to the bloodstream. And so what you see here is that, you have concentration on the vertical axis and you have time over the horizontal axis. And you see the concentration of the drug is high from the beginning because that substance is taken directly into the bloodstream and begins to get distributed. And once it gets, getting distributed, the concentration will be reducing, getting distributed to the tissues and the organs. And then it begins to get eliminated, excreted out of the body. So you have that excretion ongoing until you have all the substance getting fully excreted out of the body. So you can, compare these two kinetic curves of substances that are taken into the body by intravenous administration or by oral administration. Now, distribution. We usually refer to volume distribution, which is a theoretical, volume of a solvent in which if you dissolve a substance, the concentration of the substance in that solvent will be equal to the concentration of the substance, that same amount of substance when taken and is distributed in the bloodstream. And so you do have, so what is being depicted here is like you have some, theoretical volume of, the blood stream, the fluids in the body that you have the substance that is taken in, getting distributed in it. And then you have a certain concentration. So such concentration of the substance is what we are referring to as the volume within which you have the substance getting distributed in is what we are referring to as volume of distribution. But we saw from the beginning when we were even looking at the overview of the process of biotransformation that you do have part of the substance that is binding to some proteins or some even they bind to the cells of the tissues to have their therapeutic effect. But here what we refer to as a plasma protein binding is specifically referring to the binding of the substance to the blood plasma proteins, particularly albumin. I think you have even seen an example of the binding of, say, bilirubin, for instance. That's not an external substance. That substance is synthesized in the body, and so it's an endogenous substance. But we'll take a look at it again, and you have a reminder of the fact that even that process of bilirubin metabolism is biotransformation, except that it is biotransformation of an endogenous substance, not a xenobiotic, not an exogenous substance. You do also have term of tissue binding. Of course, that is to do with more like, the more affinity of substance for some particular tissues, the more either a therapeutic effect or a toxic effect that substance is going to be having. It also has some role to play with the half-life of the substance. How much time it takes for half of the substance to be metabolized and be excreted out of the body. And then when we're dealing with the pharmacokinetics of substances in different species, we also refer to something like a blood to plasma ratio. So that what that means is that when a substance is taken in, you have some of the substance which is bound to the blood cells, and you have some of it which is free and it is in the plasma. That's also an important aspect of it, because what it means is that the more free a substance is, as you see here from the top, the plasma protein binding, the more you have free substances, the more you have an active drug substance in the body, because a bound substance will not be active. It is the unbound substance which is free a therapeutic effect. So this is where we also have the term we refer to as bioavailability. If you take in a substance, the substance, you have some of it getting bound. How much of it is left free, which is active, is the same as saying how much of it is bioavailable. So how much of the substance will be active to have a therapeutic effect. Now, when we take metabolism of the substance, when we are looking at the main biotransformation process itself, we can have this divided into two phases, what we refer to as phase 1 and phase 2. So what you are seeing here, the two phases, what processes occur in those two phases. So you have a xenobiotic. So that's a substance that is taken into the body, by absorption. And then transformation at a stage. And there will also be elimination. Now, it is this biotransformation process which is being divided into phase 1 and phase 2. Some substances will not go through the phase 1. They go straight to phase 2, which are conjugation reactions. And these conjugation reactions too, we saw a common example with bilirubin getting related with glucuronic acid. We'll look at it again, so you remind yourself with what it is that we are talking about. Now you have also some substances that, first of all, will undergo some phase 1 biotransformation. And this can be in two ways. You have substances that will undergo phase 1 metabolism, which will result into the metabolites having still being active, but its activity may either be higher or lower than the substance itself that is taken in. So that's why we say metabolite with altered activity. Or the metabolite is inactive. That is, a substance taken in is active, but the biotransformation process makes it become inactive. So it will rather result into a metabolite which is inactive. But the process doesn't end there. Either you have that active or inactive metabolite. It still goes through the conjugation process, the second phase, before getting eliminated out of the body. So you see, the first one we looked at was that it goes straight into phase 2, that's conjugation reaction, and gets eliminated. The second one is undergoing the first phase and second phase before getting eliminated. Now, generally, these processes of biotransformation result into the conversion of lipophilic substances into hydrophilic substances so that they can easily get excreted out of the body. What it means is that the internal environment of the body is aqueous. So if a substance that is taken in is lipid, it has to be converted to a form that can easily be carried, transported in the hydrophilic environment, and at the same time can get excreted, because urine, for instance, in which a lot of the substances get excreted, is aqueous. And so the substance will have to be converted into a hydrophilic form in order for it to be easily excreted out of the body. Sweat, same thing. So all those pathways that we've looked at, those substances will have to, get converted into these hydrophilic forms. And it is this process of conjugation actually that does that. So you realize, even the example we saw when we were looking at hemoglobin, we looked at the breakdown of hemoglobin into bilirubin. You saw there was a conjugation, which is to make the bilirubin more aqueous so that it can get excreted easily out of the body. Now, if we take phase 1 reactions, these can be divided into 3 different types of reactions: oxidation, reduction, and hydrolysis. And these reactions occur mainly in the microsomes are some part of the endoplasmic reticulum. You would say these reactions occur in the cytosol, the cytoplasm of the hepatic cells, the cells of the liver. That is where you have lots of these enzymes, particularly the cytochrome P450 enzymes, which are involved in the metabolism of medicines, or let's say metabolism of drug substances or even toxic substances. And even in the oxidation itself, you have different reactions that are under oxidation. It can be hydroxylation, decarboxylation, formation of oxides, desulfurization, dehalogenation, oxidation of alcohols, oxidation of aldehydes. We'll take a look at these types of reactions. Even if not all of them, most of them we will look at it. And then reduction can be reduction of aldehydes, azo reduction of nitro compounds. And then hydrolysis will be breaking down of these drug molecules into 2 or more molecules, me particularly is to do splitting of or breaking down of some ester bonds, breaking down of amine bonds. Not limited to only this. It has a splitting of molecules into two or more smaller molecules, some hydrolytic reactions. We'll take a look at some of those examples. So that's phase one. Now phase two, reactions are conjugation reactions, which is the joining of, two or more molecules, binding of molecules, like say you have conjugation with glucuronic acid, what we refer to as glucuronidation. That's a conjugation reaction. Or conjugation with sulfates, what we refer to as sulfation. Acetylation, that is conjugation with an acetyl group. Glutathione conjugation. Methylation. So we have seen some of these reactions much earlier. And all these also occurring either in the cytosol or in the microsomes of the endoplasmic reticulum of the hepatic cells. So, I mean, if you don't even remember anything at all, just remember that you have phase one and phase two reactions. Phase one reactions are oxidation, reduction, that's redox reactions, and hydrolytic reactions or hydrolysis. And then the second phase is conjugation. All these reactions that we are talking about as phase two reaction is all conjugation. That is binding of two or more molecules to have a bigger molecule. And the purpose for that is to convert a hydrophilic substance, a hydrophobic substance into a hydrophilic substance in order for it to be transported freely in the aqueous medium and at the same time can get excreted easily out of the body. All right. Now, before we start to take like some specific substances, common substances, and begin to look at some metabolic pathways to appreciate the processes, these reactions that we've referred to in phase one and phase two. We want to remind ourselves and take a look at a specific conjugation reaction for when we were doing hemoglobin metabolism. We looked at the bilirubin, the formation of bilirubin, and that actually is some biotransformation of an endogenous substance. So this is not a xenobiotic. A xenobiotic is an exogenous substance because bilirubin is synthesized in the body. I mean, it's coming from the breakdown of the heme that is in hemoglobin and other hemoproteins, which are mainly some enzymes of the liver. And we see here, first, bilirubin is bound to blood protein, albumin. So what will you call that? That will be some conjugation, because you have bilirubin, which is binding onto albumin. What's the purpose? It's to make it flow freely in the aqueous, blood medium. And as it's flowing, it will go to the organ, the liver. So bilirubin is delivered to the liver. And in the liver, you have a conjugation reaction. Again, there is a transfer of a glucuronide. And so here you then have a bilirubin diglucuronide. Again, this can now freely flow, stored in the bile, in the gallbladder, and then secreted into the duodenum as bilirubin diglucuronide, which can then be secreted into the duodenum in the digestive system through the bile duct. And this reaction, we saw it much in detail on the right-hand side. So you see bilirubin, which binds to albumin, forming the bilirubin-albumin complex, flows to the liver, and in the liver, you can see the conjugation of the two glucuronic acid residues, which are in red. And so these glucuronic acid residues make the bilirubin more soluble. And then from there, it can either get excreted by bile into the duodenum, into the digestive system, or even in the bloodstream when it's absorbed, because it's in the digestive system. So clearly tells you that some part of it can get absorbed back into the bloodstream, but that will then go into the kidneys. And when it goes to the kidneys, the kidney will then excrete that as urobilin. And you also have that bilirubin also getting excreted as thecobilin in stool. So this process, we looked at it, but we are reminding ourselves of the fact that it's biotransformation, but it's a biotransformation of an endogenous substance. Now we can start to take a look at biotransformation of exogenous substances. So xenobiotics. And the first reaction we're looking at is oxidation. And which phase of reaction is that? Phase 1. Because the phase 1 reactions, we said they were oxidation, reduction, and hydrolysis. So oxidation is a phase 1 reaction. Now this is ethanol. Ethanol under the action of alcohol dehydrogenase undergoes oxidation to form acetaldehyde. I mean, if you look at it, this second carbon here, CH2OH, you see it is on this carbon that you have a hydrogen lost, and you are now having, instead of a hydroxy group, you now have a double bond O, and so that's an aldehyde group there. So you have acetaldehyde. That acetaldehyde can further get oxidized by aldehyde dehydrogenase into acetic acid or acetate. So that's further oxidation. The aldehyde group is now a carboxyl group. So that's an oxidation reaction by the liver. All right, so like ethanol, when ethanol gets into the body, this is the biotransformation that the liver will have on it. And of course, as it's converted to acetic acid, the next thing is that you have coenzyme A that can conjugate with acetic acid to give you acetyl-CoA. And acetyl-CoA, what will happen? Acetyl-CoA will go into the TCA cycle and generate energy. And so you see, that is even one of the reasons why persons with diabetes, for instance, are asked to desist from alcohol, because more of these acetates, acetic acid, which will be produced. It's just like if you go back to the glycolytic pathway, you realize that you have glucose that is producing acetyl-CoA. So you see glucose, which like in elevated amounts in the case of diabetes, if more of these acetates and acetyl-CoA are produced in elevated amounts, then what will happen is that you have some of these acetyl-CoA going into the formation of ketone bodies. And so even some of you are working in clinical settings, when you are doing these diabetic tests, you also look out for ketone bodies like alpha butyric acid, some beta butyric acid, acetone, or acetoacetate. These are the ketone bodies that will be formed from excessive acetyl-CoA, which will be produced in the body. Okay, somebody is asking, is this reaction reversible? So I think the reaction from acetic acid to acetaldehyde can be reversible. But what usually happens is that, so I know the reason you're asking is, for instance, maybe you have acetic acid, which is produced in the body. Will it form acetaldehyde and then come to form ethanol in the body? That's the reason you are asking such a question. There's some level of ethanol which is produced, yes, by the cells, but which is minute, not like how the concentration of ethanol that is consumed into the body. It will not be up to that. If you are thinking of it in terms of, say, yes, you can have some reversible reaction, but that level of production of this in the body is low. That's what I meant to say. It's low. And also you have with the production of ethanol using, say, yeast, for instance, that reaction will be from like a reverse reaction to form ethanol, which is used in the metabolic processes. That is like in an anaerobic condition. So I think we did look at metabolism in anaerobic condition, or maybe it is not this course. Maybe it's some other biochemistry with SLT. Also, they have something pertaining to like anaerobic condition where you can have acetyl-CoA, going into the formation of acetaldehyde and going into the production of ethanol, which is used in industrial production of ethanol. So yes, you do have that occurring, but say that is an anaerobic metabolism of carbohydrates resulting into that. All right. Now we can take some specific drugs and begin to look at, you want you to appreciate the fact that if you take in a drug substance, it isn't that a drug substance will just undergo one pathway and give you one metabolite and get excreted. But actually, a drug substance can undergo different metabolic pathways producing different metabolites. So for instance, what you are seeing on your screen now, you have this drug at the top here, which is label number 1, is antipyrine. That's the structure of antipyrine. So it's an analgesic. And you can see that it's taking different pathways. From it, you are seeing 4 arrows coming down. If you take the first arrow down this way, what I have done is I have shaded the portions of like the drug substance that are getting transformed. So for instance, if you take the drug substance, you see this methyl group, this CH3 group, I've shaded it pink. And then you see another CH3 group here, which is on this N2. So this the nitrogen that is joining to the second ring below it is N1. So if you are counting clockwise, this N is N2. Then the next will be carbon 2, carbon 3, carbon 4, on which you have the double bond O. So that's what it means. If you come down and you do same thing, that carbon, which is bonding directly on the N, is number 1, but because it's a second ring, we say 1 prime. If you're counting clockwise, the second carbon, 2 prime, 3 prime, 4 prime, 5 prime, 6 prime. So that's what it means. Now, this drug, antipyrine, if you take the first arrow, that arrow there, you see that group, which was a methyl group, has now become CH2OH. CH2OH. So that is hydroxylation. It's a form of oxidation. If you go back to what we just looked at, the types of reactions that occur, you see that is hydroxylation. That is hydroxylation, and it is an example of oxidation reaction. Now, if you come down that same pathway coming down, you see that CH2OH has now become a carboxyl group, COOH, which is a further oxidation. That's a further oxidation. For you. Now, if you take the second pathway from the original drug, again, looking at the CH3, which is on that nitrogen, you see in place of it, you now have a hydrogen, which means that methyl group has been removed. It's taken away. It's like demethylation. It's a methyl group. So that's a demethylation process which is occurring there. And so you have norantipyrine. In some cases, that process is referred to as N-dealkylation. That is to say an alkyl group. Because if you say alkyl is a residue of an alkane. So you have an alkyl group, and that alkyl group was on nitrogen. So you will see that it can be referred to as N-dealkylation, removal of an alkyl group from nitrogen. If you take the third arrow there, you see that there's a hydroxyl group which has been added on carbon number 4. That hydroxyl group was not there in the original antipyrine compound. And so that process is hydroxylation. It's hydroxylation. And specifically, you can say that is hydroxylation on the 4th carbon. And so that gives you this molecule 4-hydroxyantipyrine. There's another hydroxylation which can occur where the hydroxyl group will be on the 4' carbon. And that will give you 4' hydroxyantipyrine. 4' hydroxyantipyrine, which you see there. Now, these two molecules can go into the formation of a dihydroxy. If you take this, the molecule which is labeled number 4, which is the 4-hydroxyantipyrine, you can have hydroxylation of this 4' carbon, so that you now have 4' dihydroxyantipyrine. Or similar thing can happen if you already have the 4', getting hydroxylated. Then you can have the carbon number 4 getting hydroxylated next. So it's either you have 4 getting hydroxylated first before the 4' gets hydroxylated, or you have the 4' getting hydroxylated first before you have the 4 getting hydroxylated. Both of them will give you the same thing, which is 4,4' dihydroxy antipyrine. So we can see one drug substance is undergoing different oxidation reactions. At the same time, we also saw the process of dealkylation also ongoing there, because that dealkylation, I think, is also one of the oxidation processes that we saw. So the N-dealkylation, we can see it much more here. We're seeing a drug substance, codeine, which is also a common component of painkillers, some strong painkillers, analgesics. And here again, you see codeine getting converted to norcodeine by the process of N-dealkylation. If you look at this CH3, which is shaded here on the nitrogen, this is removed when you look at the structure of norcodeine. That is removed from there. And so that is N-dealkylation. If you look at the pathway going up, codeine getting converted to morphine. You see the CH3, which is on that oxygen there, shaded pink. You see it's removed in morphine. And so that process is referred to as O-dealkylation, because the alkyl group is being removed from oxygen. That's why it's O-dealkylation. Same thing if you have morphine getting converted to normorphine. See the CH3 there again on nitrogen is removed. And so this process is also N-dealkylation. So, I mean, this is how you pay attention to which functional group there is getting converted. So you can have some big molecule, large, no matter how complex it is, you just have to focus on these, some functional groups and the differences between two molecules. And then you can tell the process which is ongoing. Of course, these are one of the examples to, I was referring to the fact that some drug substances that are taken, their metabolites can be, more toxic than the substance itself. So, for instance, norcodeine is more toxic than than codeine. Similarly, normorphine and even morphine is more toxic than codeine. About I think norcodeine about maybe some 3 to 5 times or so. Normorphine maybe up to about 6 times more toxic than the codeine itself. I don't remember the specific ratios, but I know what I remember is this metabolites are more toxic substance itself. All right. Now a very common one, paracetamol. Take a look at paracetamol. Now this is paracetamol here. The structure. Acetaminophen. So that is N-acetyl benzoyl. You have different pathways here. That the interesting thing is, this drug paracetamol, look at it. You have 50 to 70% of paracetamol undergoing glucuronidation. That's the similar reaction that we saw with the bilirubin binding to glucuronic acid residues. Similar reaction. So here you see that's glucuronic acid there. And glucuronic acid is binding on in place of that hydroxy group of paracetamol, glucuronic acid is binding to paracetamol. So what it means is that 50 to 70% of paracetamol that is administered just gets conjugated with glucuronic acid and gets excreted out of the body. That's what it means. And you see here about 25 to 35% of paracetamol when taken undergoes sulfation. So you see, in place of the hydroxy group, you have a sulfate group there in paracetamol sulfate. And that is an inactive metabolite of paracetamol. You see, 5 to 15% of the drug. And that's where, so here you are seeing one of these cytochrome P450 enzymes, which is catalyzing that oxidation reaction there. I mean, there are some enzymes also here, SALT1, A1, SALT1, all these sulfation enzymes. There are some transferases, just like the glucuronic acid, the glucuronide transferases as well. And you see that this pathway, this oxidation, which is converting 5 to 15% of paracetamol to this N-acetyl paraquinone. In fact, this particular molecule is the toxic metabolite of paracetamol. But if paracetamol is taken within the right dosages, this toxic component, the toxic metabolite which is produced, can gets almost immediately gets converted. It gets conjugated to glutathione and then gets excreted out of the body. This can get excreted by conjugation to glutathione. But if in the case of paracetamol overdose, what it means is that you have an increase in the production of this N-acetyl paraquinone, which does not get quickly converted, getting conjugated to this glutathione. So what will happen is that this toxic metabolite begins to bind to proteins, forming what we refer to as some protein adducts, and that can result into some inflammation in the liver and in other organs. And in fact, in very high dosages, this can result into fatality. So paracetamol overdosage can result into paracetamol toxicity. And now you see the pathway that gives you that therapeutic effect of the drug. Look at it. You just have 1 to 2% of paracetamol undergoing deacetylation. First of all, that is removal of the acetyl group there. And so you now just have an amine group. So you have a para-aminophenol. And it is this para-aminophenol which will then bind to arachidonic acid. That's a fatty acid with, I think, 20 carbons. And it is this arachidonyl aminophenol that is active. It is so what is usually named AM404. That is the active form of the paracetamol that has the analgesic effects, the pain relief effects that is experienced when paracetamol is taken. So you see how interesting this can be, that you just have 1 to 2% of the drug that goes into that pathway and has its effect. The rest of it, or most of it, is just getting excreted as metabolites. Now you can have some other drugs which are like, what we refer to as primary amines. These are some benzene rings with primary amino groups, terminal amino groups. And these can undergo processes of acetylation. Acetylation is conjugation. So that's a phase 2 reaction. And so you can have, on that amine group, you now have the acetyl group binding, and you have an N-acetylated metabolite. The coenzyme is cleaved off. And so you have a coenzyme A there, and you have the N-acetylated metabolite. Now, the N-acetylation actually also has, some genetic polymorphism to it. There are some people who are fast acetylators, and some are slow acetylators. And so that's, this brings us to what we refer to as personalized medicine. I think we'll take a look at it as we go ahead. Now we've gotten to excretion of the drug, what you're referring to as elimination. And you see you have different routes by which the drug is eliminated. Drug which is eliminated through urine, we refer to as renal excretion, where you have, the kidneys, where urine is formed, and then that urine is stored in the bladder, and then is then excreted. You have, the drug substances, digestive system that can, get excreted with stool. All right, so let me see. Bright Nate Saki says all these drugs we take in, where some are converted to toxic, though would be excreted. Our kidneys not affected. And so you see, that is why there is dosage which is written on the drugs, how the drug should be taken, the dosage. If it's taken above that, that's when you start to have, some toxic effects. So you see that every drug has some dosage that has to be taken. Even though on some it's written that even at high dosages, when they were carrying out trials, maybe animal trial and maybe even clinical trials with humans, no toxic effect was seen. But once the therapeutic dosage is established, then that's the dosage that you should be taking in order have the effect you are referring to. So your liver, the kidneys, they get affected when you take overdosage. That's what it means. All right. Then you also have what we refer to as biliary excretion. We refer to the fact that some of these medicines, after they get metabolized in the liver, they get stored in the gallbladder with bile, and so they will get excreted with bile into the digestive system. That is what we refer to as biliary excretion. Some substances, we saw that they get excreted through exhalation, getting excreted, through the nostrils as gas. That we refer to as pulmonary excretion. Like the ketone bodies that we refer to, acetone, for instance, is a volatile gas. It's a volatile liquid. You find it in the lab, which easily gets exhaled, and you actually experience the smell of acetone from excessive alcohol consumers, for instance, where they have these ketone bodies being produced a lot, and you have acetone getting exhaled with the exhaled air. You smell a pungent sort of a smell from them, and you don't want to go close to them. That kind of excretion is what we are referring to as pulmonary excretion from the lungs. Dermal excretion. So that's what we said. You have excretion through sweat. So you have the sweat glands that are producing sweat, and from the sweat duct coming to the surface, you have some of the drugs getting excreted in that manner. Lacrimal excretion. This is particularly for, say, eye drops, nasal drops, ear drops. You have the visual canal, the buccal cavity, that's the mouth, the ear, the auditory canal. All of these connect, somewhere in the throat, which is why sometimes if you take some nasal drop or some eye drop, you taste it later. Like you feel it, you taste it in your throat. And so you have some of these drugs also being, for instance, this particularly, say, the eye drops, or even the other ones that I mentioned. What can happen is you can have the excretion of them, coming from the lacrimal gland, the gland which is, responsible for the production of tears that come into our eyes. So some drugs are excreted through there as well, what we refer to as lacrimal excretion. And that's the tears too is not only coming from the eyes, but tears, you see, you can pass through the nose coming out as well. All right. You do also have salivary excretion. So some drugs taken in can get excreted through saliva. Because the glands we are referring to, blood flows into them. So once you have these drug substances in this blood flow to these tissues, some of these drugs can get excreted from these glands as well. So you have salivary excretion, and you do have some drugs in lactating mothers. Sometimes some drugs are prohibited, say for lactating mothers, that if you are a lactating mother, you shouldn't take them, because, and even sometimes if it's not known, it's not clear whether the drug is excreted in breast milk or not, because it's not known, the best thing is not to take it at all. So you find that you have some of these substances that are also excreted in breast milk. Now, we can take a look at some other aspect of biotransformation in terms of the environment. So this time it's not transformation in the human body, but outside, which is also something pertaining to health. So you have some herbicide. This is trichlorophenoacetic acid. What is label number 1? The name is below here. I've written it there. 2,4,5 trichlorophenoacetic acid. And this herbicide, actually was used in the Vietnam War by the American soldiers. They sprayed it with a helicopter over large fields, because there were some forests covers that there are some Vietnamese soldiers that were, they use these forest covers as their hiding places. So the soldiers decided they use this herbicide on those forests, and then so that the place becomes clear and there will not be anybody that can be hiding in those places. And this herbicide was actually used, I think in the 1940s up to about maybe 60s thereabout before the realization came that what happens is that in the environment, the substance undergoes a hydrolytic process. So you see the acetic part breaks off. You have that acetic acid, and you are left with the trichlorophenol. And this trichlorophenol actually gets conjugated. So two trichlorophenol can bind and form what we refer to as TCDD, the tetrachlorobenzodioxin, which is a toxic, which is a toxic substance. It's persistent in the environment. It does not easily get degraded. And then this TCDD is it can even affect the fetus, pregnant women when they breathe in these things, affects the immune system, the nervous system. So it's a very persistent toxic substance. And so now this herbicide is banned, is no longer being used anywhere in the world. You have it banned. And in fact, this dioxins are some of the persistent poisonous substances that you can find in the air. That is why it's very strange that currently in Ghana you still have places like Agbogloshi where they are burning things like this recycling stuff at the center of the city, and everybody seems to just be okay with it because it's clear these things are very poisonous substances that go far to affect whole populations. So the same way that there is so much of talk about the destruction of forest cover and the toxic chemicals which are being used in this, mining in the regions. That same emergency should have been attached to something like burning of things. It's strange. Like people just take some rubbish and somewhere in front of their house and start burning, things like that. Those are very dangerous things, exposing ourselves to a lot of these dioxins. So, in fact, there are a lot of things that are overlooked, which really result into, so many health problems in the environment that you now, as you are getting trained as a medical science, specialists, you should pay attention to some of these things. I mean, from now on, you shouldn't be home and just start burning anything. Just pack something somewhere and just set fire to it. I mean, you should advocate there to stop some of these things because they're actually really dangerous. There are some foreigners when they come to Ghana, they get very surprised. And it's so shameful to just see people just burning anything and everybody's looking at them. And even at the center of the city, sometimes even around our university there, you see some of these things happening. And nobody seems to really bother about it. All right. We have another one here, parathion, which is a pesticide also, which was used, but is also now banned. Parathion. And you see parathion also when exposed to light undergoes photooxidation to paraoxone. It can also undergo hydrolysis into paranitrophenol and diethyl phosphorothioate. And the photooxidation to parathion resulting into paraxone. That paraxone is even more toxic to humans, mammals, than the parathion itself. also, acting on the central nervous system. So it can result into some paralysis in the end. So this is also banned now. We have so this has some history, some research history behind it. That you have phytosterol. These are some steroids in plants that in the environment, some microorganisms can act on this phytosterol, converting it into male sex hormones, androgens, testosterone. It's a form of oxidation. You can see clearly that this hydroxyl group here is now, this cyclic ketone group. And you see some in one of these states, I don't remember whether it was California or so, you had there was a factory which was situated along a water body, and that factory was producing they were processing nuts. And you would think that nuts, they are plant products that you don't expect to have any toxic substance from, because it's food. If anything at all, if you have this released, like the waste released into, say, water, it will become food for say the aquatic animals to feed on and things like that. Only to realize that along that water body, some fishes were noticed, the female fishes were noticed to develop masculine features and were even becoming sterile. And so when that effect was traced, it was traced to that particular factory that was producing these nuts. They were processing these nuts and releasing the waste into the water body. And whilst you have this waste which was being moved down the water body, you had phytosterol, which was in abundant amounts in these nuts, getting converted by some microorganisms in the water into androgens. And so you had these male sex hormones now acting on these female fishes and having such an effect of masculinization of these fishes. So you see how you can have biotransformation. You have a biotransformation of this harmless substance, phytosterol. Even the sex hormone itself, androgen, is harmless, but you can see the effect that it's having on organisms, aquatic organisms, which is to say masculinization of these female fishes and making them sterile. Now, we also see cimetidine is a drug substance which is used for the treatment of ulcers, or peptic ulcers, heartburns. But the side effect of it is that it reduces gastric acidity. So that gastric acidity that it is reducing, it creates the side effect of now a condition in which bacteria and fungi in the gastrointestinal tract get, they have that environment for more proliferation, more growth. And when that happens, you have food containing nitrates, even drinking water, some beverages, and you have this growth of bacteria and fungi, now converting these nitrates into nitrites. And with further, bacterial action on these nitrites, there is now the formation of, what we refer to as nitrosamines. And these nitrosamines, can combine with some thiocyanates, chlorides, also coming, from food substances, with also, the activity of this bacteria on these nitrosamines, that is nitrosation. And in the end, what happens really is that you have an increased risk of gastric cancer. So on one end, the drug is being used to treat peptic ulcer, heartburn. And then the side effects, if the drug is being taken over a long period of time, this can result into this increased risk of gastric cancer, because once you start having the production of these nitrosamines, you have this fecal bacteria, which will increase the carcinogenicity of these nitrosamines that are being produced. That will also result into some inflammation of the gastrointestinal tract, the damage of the stomach lining cells, the mucosa, the mucous lining of the gastrointestinal tract, is damaged, and that will cause the digestive tract to be more susceptible to inflammation, which will then result into this, increased risk of gastric cancer. So here again, you see, is biotransformation, cimetidine, that is being acted upon by bacteria and fungi, which is resulting into that. Now, I mentioned that when I was talking about the N-acetyltransferases, I mentioned that you do have some genetic polymorphism, so that in different groups of people, you have some people who are fast metabolizers, some are slow metabolizers. So this is an example of a drug, isoniazid, which is an antibiotic used for the treatment of tuberculosis. That you have a research done, over, say, 267 volunteers, that you had the concentration of the drug in plasma of such persons over, a certain period of time, and you see the distribution of the persons such that you have, in some persons the concentration is very high, and in some persons concentration is intermediate, and in some the concentration is very low. And what it means is that drug, isoniazid, when it's taken in some persons, is getting accumulated, and so you have the concentration being very high. And intermediate metabolizers will have some, medium concentration with them, and the very fast metabolizers. So they are metabolizing the drug very fast, and the drug is getting excreted out of the body. You have them having the concentration being very low. I mean, this brings to the concept of personalized medicine, so that it's not the same dosage, which is the same dosage of a drug which is given to everybody. Because if you give the same dosage to everyone, like what we are seeing here, in some of the people, that substance, the drug substance, if it's a fast metabolizer, maybe the same dosage you are giving will not have the therapeutic effect that is intended. And if it is in a slow metabolizer, that concentration will rather be resulting into a toxic effect after it's getting accumulated in the system. And perhaps the intermediate, yes, it will be working for them, which means you should first determine that genetic disposition of the person before you give particular dosages. So we say personalized or it's customized dosage so that the person will, the intended therapeutic effect will be achieved, and there will be elimination of any toxic effect in the end, at least reduce any toxic or adverse effect of the drug in the end. All right, so I think this is where we'll end. I think we still have like some 19 minutes more. So if there are any questions, any contributions, anything anyone would like to say, you can write in the chat or you raise your hand. I will enable your microphone and then you can unmute yourself and speak. The floor is open.